Let's go ahead and get ready to start. Welcome to this afternoon's webinar on the NHGRI-EBI Catalog of Genome-wide Association Studies, or the GWAS Catalog. My name is Joanella Morales, and I work at the European Bioinformatics Institute, the EBI, and I'm one of the curators on the GWAS Catalog team. And what I hope to do in the next 30 minutes or so is to give you an overview of genome-wide association studies in general. Then I will talk about the catalog itself. First, I'll provide some background. Then I will look at the content that we have in the catalog. Then we'll discuss how to access all the data. And then hopefully we'll conclude our time with a hands-on exercise to make sure that you are familiar with how to use the catalog. Okay, so GWAS stands for Genome-Wide Association Studies. And the main goal of a GWAS study is to identify genotype-phenotype associations. We know that changes at a single position can affect phenotype. This slide here shows a very simple diagram where you can see that different alleles highlighted here in the circle can have a different impact on a phenotype, in this case, eye color. Now, this is a very simplistic diagram because we know that eye color is affected by probably many genes, and it has a strong environmental component. But nonetheless, the goal of a GWAS is to identify these changes at a single position that do have an impact on the phenotype. Now, some key concepts about GWAS to remember. GWAS is a hypothesis-free approach. It scans whole genomes for genetic variants associated with a given trait of interest. GWAS are particularly useful to find variants that contribute to common complex diseases, for example, diabetes, or heart disease, for example, cancer. Now, this approach has already achieved considerable success. A vast number of variants have been identified, and many traits have already been surveyed. In fact, I hope you can see right below here where I've presented the most recent stats from the GWAS catalog, from the release that we did this week, that the catalog already contains about 3,000 publications and more than 37,000 unique SNP trait associations. So definitely the GWAS approach has been successful. But one thing to remember about the GWAS is that these associated variants that you find are not necessarily causal variants. They, in fact, often merely tag an associated region of the genome for further study. So, for example, if you've done your GWAS and you've found many variants that are associated with your trait of interest, that doesn't mean that all those variants are causal. It just means that you found regions of the genome that you should then go and arrange for further studies to determine if you can find which one of those variants actually causes the trait of interest. This slide basically shows you the typical GWAS study design. Here you have your cases and your controls. Cases are individuals that obviously have either the disease or your trait of interest, and your controls are individuals that are unaffected, that don't have the particular disease. Usually your study starts with a genotyping of an array, and more than 100,000 SNPs are usually analyzed in a particular GWAS study. And the key of a GWAS is to look for variants that have differences in allele frequencies between cases and controls. So if you look at this diagram here, I'm illustrating this by showing you one particular variant, RS1234, carrying the A allele. And you can see that the frequency of this allele in cases is 58%, whereas the same SNP and the same allele in controls is only 16%. So what you're looking for are SNPs that are different that have different frequencies between cases and controls. Usually a statistical analysis is performed to determine the level of significance of that difference between cases and controls, and this data is all summarized or visualized in a particular display. In this case is called a Manhattan plot. Here on the y-axis, you can see the level of significance of your association, and on the x-axis, you can see all the variants that you have identified in your GWAS organized by chromosome. And what you can clearly see in this example is that our SNP that we were looking at, RS1234 is very significant. In fact, is the most significant in this particular study. Now, I've already said that the GWAS approach has been successful, that thousands of SNP-trait associations have been identified. But then the question is, how and where can I find this vast amount of data? Do I then need to go and read 3,000 papers? Well, this is where the GWAS Catalog comes in. Now, let's go in and take a look at the GWAS Catalog itself. The NHGRI-EBI GWAS Catalog, that's the full name of the catalog, is, as its name implies, a catalog of GWAS studies. It's a publicly available resource. It was founded by the NHGRI in the States in 2008, but since 2010 it has been a very fruitful collaboration between the EBI and the NHGRI. The aims of the catalog are very simple. It's to extract data, GWAS data, from published genome-wide association studies through a process of manual curation, and then make that data freely and publicly accessible in a search interface or in the GWAS catalog. Now, for a study to be eligible for inclusion in the catalog, it has to be a genome-wide study. That means that you're looking for variants all across the genome. It also has to be array-based. That means that your genotyping was done using arrays, and more than 100,000 SNPs must be analyzed for the study to be eligible. For an association to be eligible for inclusion, it has to be significant, which means that the p-value has to be at least 1 × 10⁻⁵. And the SNP that we actually extract or include in the catalog is the most significant SNP at each independent locus. So we don't extract all the particular SNPs, we extract the one that is the most significant at each distinct locus. So now let's look at the catalog. What kinds of data do we include in the catalog? Well, this shows again your typical study design. And we include information about the publication. So for example, the journal, the authors. We include the date of publication. We also have information about the participants in the study. So we might include what phenotype or trait we are interested in. There might be information about the size of the sample that was included in the particular study, and information about the ancestry of the participants. We also extract data about the genotyping process. So for example, the name and make of the array that was utilized in the study, and also the number of SNPs that were analyzed in the GWAS. And finally, we extract information about the results that were obtained. And this should now look familiar. This is your typical Manhattan plot. Now, looking in detail at the results that we extract, we have variant information, so the SNP identifier with its RSID. We also extract information about the allele that is affecting the phenotype or the trait. We look at the closest gene to the particular variant, and then we include information about where the variant sits on the genome, so its chromosome, its position, etc. In terms of the association itself, we extract information about the strength of the association, so the p-value. We also extract information about the effect on the trait. So usually it's a beta or an odds ratio that will describe the impact of the SNP on the trait. And then we extract information about the trait that has been analyzed in the study. Now, this is good amount of data. How do we access it in the catalog? Well, there are multiple ways that you can access data in the catalog. The most common way is through the search interface, and I'm going to go in detail into what each of these ways in a minute, but I'm just showing you generally that there are several ways of getting the data. Through the search interface, this is the typical, most usual way. Through a complete download of all the catalog data through a tab delimited file. So you can download the entire catalog, you can open it in Excel, and then you can do any kinds of analysis using your Excel spreadsheet, for example. But we also have an interactive karyotype diagram that also serves as a portal into all the data that's included in the catalog. Now, we know that there are some users that prefer to access the data programmatically, and we are working very hard to provide programmatic access, and that will be coming soon, very soon. So let's look at accessing the data using the search interface. Now, first, let me say a few words about how the search actually works. So the search provides returns exact string matches within data fields. So if you're searching, for example, for type 2 diabetes, it will look for that within the publication information. So if it appears in the title, it will identify studies that have your term in the title, or if it provides the information, you will look at the field in your sample descriptions, or in your genes, or in your SNPs, or in your trait. So it's a very comprehensive search that the catalog does through all of the data fields included in the catalog. But it's also very powerful because it's supported by an ontology. In this case, the ontology we use is the experimental factor ontology, and I will just very briefly, for those of you who are not familiar with ontologies, the purpose of it, or one of the benefits of it, is that it provides structure. It gives structure to all the data included in the catalog, and it also helps to define relationship between the different terms included in the catalog. So for example, consider, for example, this list of traits related to diabetes. Now we could have a search where you could have a list of all the traits as included here, but this list doesn't give you any information about the structure of the data or the relationship between the terms, all of these terms. However, once you integrate all this information into an ontology, you can then see that diabetes is actually a metabolic disease. You can see that there are 3 subtypes of diabetes. You can see that diabetes has other terms that are related to it, and this gives you structure and it allows you to do a more refined search. Now specifically, how do we search the catalog? This is what the search interface looks like when you access the data, when you access the catalog using your typical URL. You can put in the search box that I have put in red. You can put any text of interest. But you can also click on your search icon, and that will take you to a search-specific page where you can still put in your term of interest, but you can also now look at all the recent studies in the catalog. You can browse the full list of available terms, and you can do your search that way. I am now giving you an example of doing a search using type 2 diabetes as our trait. And here, again, as a reminder, you can search by trait. You can search by SNP, so you can provide a specific RS identifier, for example. You can provide a gene, your gene of interest, an author, a PubMed ID, or a region. And the search looks into all of those fields. Now, when I type 2 diabetes, it is searching obviously by trait. And here, the results will be displayed in 3 tables: a studies table, an associations table, and a traits table. Also, you can see some quick stats about your search. So for type 2 diabetes, the interface was able to identify 92 studies, 777 associations, and 43 traits related to your type 2 diabetes trait. I am now going to show you each particular table so you can see the kinds of data we display in each one. Now, your studies table provides information about all the studies in the catalog related to your search. So here I search for type 2 diabetes, and you can see information about your author, your publication date, your journal, your title, and how many associations have been found in that particular study. This table is interactive, it's sortable, you can do further searches, it links out, for example, to PubMed ID, through the PubMed ID to PubMed, but you can also see very specific information about the sample given in each study, and you can do that through an expand button that I have here shown in red, or you can expand all the studies that are included in your table. To illustrate the point, I've just expanded this one study with the author of Cook, published in 2016. And you can see that we organized the sample by stage of the analysis. So if there was an initial study, that is described as initial, and if there was a replication stage, that is described as a replication. And you can see that there's information about the sample size, about the ancestry of the participants, and also there's information about the platform. So in this case, no array information was provided, but we know the number of SNPs, and we know that there was imputation that was carried out. To zero in a little bit more about the ancestry in the catalog, and I'm spending a few minutes on it because I know this is an area of interest for many users. How do we capture ancestry in the catalog? Well, we do so in two forms. We have a free text description, which is basically a text using the information provided by the author about the participants. And this is here highlighted in this yellowy orange color, where in this case, the initial stage, we've described them as 8,000 European ancestry individuals. And that's based on information provided by the author. But we also capture the data in a more structured format using a defined number of terms. And the purpose of that is to, again, provide structure to the data to make sure that our descriptions are consistency across the catalog, and that all will improve the searches that the user can do. So, and this is done by mapping or taking all these free text descriptions as presented here and assigning them to one category as we have defined in the catalog. And very briefly, these are the different categories, ancestral categories that we include in the catalog. And so what we do is we take each sample as described by the author and we assign it a category from this list here. Moving on to the associations information. This is all presented in the associations table. Very much like the studies table, it's interactive, it's sortable, you can do further searches, you can link out. And what I am showing here is that you can see information about your SNP, so your RS identifier, and you can see the allele that is conferring risk. So in this case, increasing the risk for type 2 diabetes. I have circled this in green just to show that by default, we present the data in the associations table ordered by strength of the p-value. So you will always see your strongest association first. There's information about the effect, so it's either an OR or a beta, but there's also data that we obtain through Ensemble, and this is what we call mapping information. This is additional information about the particular SNP based on information already included in Ensemble. So the region, the location on the genome, the class of SNP, and the gene that is the closest to the SNP of interest, all of that is obtained from Ensemble, and it provides the user additional information about their SNP that they have identified or the SNP that is associated with their disease of interest. The third, if you're interested in finding out more about your SNP, you can link out to Ensemble to see all this data and additional data that we have in Ensemble. For example, that will link out to a SNP page that gives you additional information about population genetics or any other information that might be of interest to the user. The final table is your traits table. Much like the other two, it's interactive, it links out. You can link out to publications or you can link out to your mapped traits. And what I'm illustrating here is that because of the ontology, and as I mentioned, the ontology gives you a richer, more robust search, you can see your traits of interest. For example, if you search by type 2 diabetes, it pulls out type 2 diabetes, that's your trait of interest. But it also pulls out some traits that are related to type 2 diabetes. For example, fasting plasma glucose is definitely related to type 2, while it's not type 2 itself. And you can also pull out other studies that are related to type 2 in that the sample, the participants in the study were all affected by your disease of interest. So now this shows you how using the ontology gives you a more comprehensive search. Now, let's say you are not interested in seeing sample backgrounds or any other related traits. You're just interested in your trait of interest. We do offer the ability to filter your results, for example, by trait, but also by p-value, by odds ratio, by effect, by date, or by trait. And here I'm illustrating what I just mentioned. If you do a search by type 2 diabetes, you'll find 92 studies. Some of those are type 2, but are also other things that you might not be interested in. You can then filter so that you only see type 2 diabetes, and that's illustrated in the right-hand panel, where you can see that now your traits have gone down to 1, the one of interest, and your number of studies and associations are now more specific to your trait that you've searched for. All of this data is, of course, downloadable by clicking on this link here, and then you can do further analysis using tabular formats. Now, let's say that you don't want to do a fully comprehensive search using the interface. You just want to access all the data and analyze it that way. So you can download the entire catalog using one of our downloadable or all of the downloadable spreadsheets we offer. Here you can see files, you can see the diagram and summary statistics that are all things that information you can download. But in particular, your spreadsheets, you have an association spreadsheet, a studies spreadsheet, and an ancestry spreadsheet that provides all the information included in the catalog. And we also include accession identifiers to help you connect the 3 spreadsheets through looking at these accession identifiers. So if you're interested in one particular study with its accession identifier, using that you can find the data in all 3 spreadsheets. You can also access the data through our catalog diagram. This is an interactive diagram using karyotypes. That means that you use and you visualize the data on chromosomes. Each dot represents a different association, and here you can see so many dots. These are all the associations with a level of significance of less than 10 times 10 to the minus 8, and the color represents the different types of traits. But let's say you are interested again in type 2 diabetes, you can then filter your diagram such that you only see your dots of interest. And then if you hover over a particular dot, you can then see SNP specific information. You can see your RS identifier, you can see your p-value, you can see your study, and then you can enter the catalog using the karyotype. Another type of download that we have recently made available are all the summary statistics. So as I mentioned earlier, currently we only curate and extract the most significant association from each locus. And that's only if it meets our p-value cutoff of 10 to the minus 6. And that's what I'm showing here with these red squares, that the top SNP at each locus is the one that's included in the catalog. However, having all the association data from a given study is useful because it helps identify co-located signals across studies, and it also increases the potential for downstream analyses. So we have now, for studies that provide summary statistics in their publications, we are now also making those available through the catalog. Finally, as I mentioned before, many of our users don't want to sit and look in a search interface. They don't want to deal with Excel. They just want a programmatic way of accessing the data. At the moment, we provide stable URLs that can be of use, but we now also have all our data, our archived versions on an FTP. But what we know that users really want is to access all the data programmatically. And so we have now released in beta version our new REST API that allows programmatic access to all the data. Now again, as I just said, this is in beta version, and we do need people, users, who can help us test it. So if any of you are programmatically minded and would like to test this new functionality, we recommend and suggest that you please contact us at GWAS Info and let us know of your interest. The final slide, just showing that, as always, we are very interested and keen on improving our resource. Many of these improvements are based on feedback that we've obtained from users. We are interested in making information more interactive and more user-friendly. So we are in the process of designing pages that are specific for each variant or for each trait or for each study, so you can see additional information. And we also want to improve our search ability, for example, using batch searching. And we also want to extend the scope of the catalog. As I mentioned, at the moment, for a study to be eligible, it has to be genome-wide. And we're now interested in including non-genome-wide data, for example, exome array data or targeted array data. We also want to expand our training, and I know in the end there will be a survey so you can comment on this particular training, but we are keen on expanding our resources. We're also keen on figuring out how to prioritize our curation, and I would point out that we have a survey where you can just go and let us know what kinds of data do you most like to see. Do you want to see data with a large number of individuals or with significant p-values? All of that you can go and let us know through this survey. We are very active on Twitter. We also have an announcement list where you can sign up to get updates on the catalog, and you can always contact us using our GWAS info at ebi.ac.uk. Last but not least, these are all the members of the team. We have a good mix of individuals here at EBI as well as in the States at the NHGRI. We have developers and curators, and it has been a very fruitful collaboration. I'm very happy to be part of this team. So now, with the time we have left, it would be good to get on and do some hands-on exercises. You can also, I know there's an opportunity to ask questions at the end, but I thought it would be useful if I illustrated how we can use the catalog to do our searches. And so this is one particular case study. I'm going to walk through question 1. If we have time, we'll look at questions 2, 3, and 4, but if not, maybe I can say that's some homework for you to take home and try it on your own. So the question here is, which regions of the genome are most strongly associated with breast cancer? Second question is, which SNP has the most associations with breast cancer? Is this particular association ancestry specific, and is it breast cancer specific? So I will now share my screen, hopefully successfully, to show you the catalog live. And here is the entryway at ebi.ac.uk/GWAS. This is a GWAS catalog. I think you have seen now this several times in this presentation. You can search, you can look at the diagram, you can download, you can see all our documentation. There's frequently asked questions, there are abbreviations, other information about how we do our curation, and that's all in this documentation. You can also find it in these tabs up here. And then coming soon, there's additional information about the ancestry data in the catalog. Right, so the first question is, what are the significant regions of the genome that associate with breast cancer? So one way to do that is to simply type cancer or breast cancer. I will start out with cancer. And hopefully you can see that there's many studies, associations, and traits. So obviously looking down our list of studies won't be the most helpful. This is where it is good to have this filtering tool. So then you can come down, you can click on your reported trait, and thankfully the first one on the list is breast cancer. You can click on that, but you can also look and see that there are other breast cancer related terms that you might be interested in. For example, there is breast cancer estrogen receptor negative. There's breast cancer survival. So this is for the user to go in and determine how they would want to use the filtering tool. But in this case, I'm just going to select breast cancer. Then you apply your filter, and that brought down your studies to 33 studies, 222 associations, and the one catalog trait that you looked for, which is breast cancer. And that should be very obvious here. You see in your reported trait, everything is associated with breast cancer. You can see the number of associations for each study. Let's say you are interested in reading the most recent study. That is how it should appear by default, but you can also sort. You can see the oldest study was published in 2007, the most recent in September of 2016. That's included in the catalog. Obviously, this is a manually curated resource, so we are actively curating, and it might take a few weeks between the paper being published and it being included in the catalog. If you're interested in reading this publication, you can click on this, and now it takes you to Europe PMC. You can read the entire text by clicking here, or you can read the abstract to determine if it's something you want to look at in more detail. And then go back to your catalog. And now the question we had was, what are the regions of the genome that are most associated with breast cancer? Well, it makes sense then to go to your associations table and see which SNPs have the strongest p-value, which would be presented in this p-value column. As I mentioned earlier, by default, the associations are presented in order of p-value, the strongest p-value, and so that's what you see here. Now, you see there are different regions. There's this SNP is in 10q, this one in 16q. So to find the regions that are most associated, you could then move on to download your association results, and which appears in tabular form, in this case as a .ts. Oh yes, sorry. I need to show you the screen where you actually see. So you can see the download as a TSV file. If that is not of interest, you can open that as an Excel you can open that using Excel, and you can open it with Excel here, which is what I'm doing. Let it open. And yeah, right. All right, yes. Great, and so now you can see all the associations that are related to breast cancer that I have. Oh, wait a minute. And you can then go ahead and filter in whichever way you would like to filter. So in this case, we wanted to filter by p-value. So you can sort by p-value. And you will find the associations that are more significantly. You can find that here in your p-value column. Gives you the strength of the association. You can then look at your SNP of interest, which is presented here. You can look at your region. And one way that you could get at the regions, you could just list, these are the regions that are more significantly associated by strength of p-value. But you could also then go further and sort by RSID. And you can see if there are any RSIDs that are that have been validated, that have been extracted more than once, that have been, and for example, the first example here, you see that this particular ID has been, this particular association has been identified in 4 different studies, which gives you a sense of validation that it has been identified more than once. So this is how a simple way that you can use the download spreadsheet. There are many other uses that you can have for the spreadsheet. You can filter and download and do more formulas to find the information you're interested in. So I have now shown you how to use the spreadsheet. I have shown you how to do the search using your search interface, but now we can also go to the diagram, and this diagram, you can make it bigger or smaller. You can move it around on your screen, much like a Google Maps would. And here, again, if you're interested in breast cancer, you can filter by breast cancer. And now you see all the variants that have been associated and their position on the different chromosomes. You can zoom in. You can see that in further detail, for example. And it's purple because purple are all the cancers. So there's about 877 associations with cancer in the catalog, and you have now filtered for the 95 that have been associated with breast cancer. Let's say you are very interested, let's say you knew a region of the chromosome that you were interested in. For example, chromosome 9, which is this one here, or chromosome 10. You can zoom in. And now if you hover over your circle, which represents a particular SNP, and you click on it, you can now see the information about the SNP, the information about your p-value, your trait, your EFO mapping, and your study of interest with a link out so you can go and read that paper when you have time. And we, of course, are looking for ways to make all our diagrams and our tools more user-friendly. So if anything that I have shown here makes you think, oh, it would be great if it did X, Y, or Z, I would encourage you to again, gwasinfo@ebi.ac.uk and let us know what we could improve or how we could make it better. I have now gone through our very simple case study and shown you how you can use the catalog. The rest of my slides are basically showing you the same information. Well, thank you very much for joining me for this webinar. I hope it has been useful. And again, if you have any questions, you can contact us at gwasinfo@ebi.ac.uk. You can look us up on Twitter, and feel free to let us know if there's anything else that we can do to improve how we present our data in the catalog. Thank you very much. Goodbye.